with the plant respiration part. And in plant respiration part, in last class, I told you it was the summary of the respiration, right? It was the summary of the respiration. And in the summary of the respiration, I told you that either it's a plant or it's animal, the respiration takes place in the similar way, and same kind of the cycle goes on, right? So the glucose reaches to the inside the cell, right? It is taken by the cell, by the plasma membrane, and after reaching into the cell, now the burning of glucose starts. But for the glucose cannot burn directly; it have to convert into some other compound. Actually, glucose cannot enter directly into inside the mitochondria. Mitochondria is that furnace where it have to be burned, right? But it cannot enter inside the mitochondria because its size is larger. So it have to change its size. So what happened? A cycle goes on that is called glycolysis. So glycolysis cycle goes on in the cytoplasm part. So when glycolysis cycle goes on, then this glycolysis cycle, uh, formation of ATP takes place, means 8 ATP are produced in the glycolysis cycle. Then by the glycolysis cycle, the 6 carbon compound changes into 2 molecule of 3 carbon compound. That is a pyruvate. Now the pyruvate is a smaller molecule. It have capability to enter inside the plasma membrane, inside the membrane of mitochondria, right? Biomembrane of mitochondria. Now the pyruvate entered inside the mitochondria into the matrix part of the mitochondria. There is a gel-like substance inside the mitochondria, that is a matrix. But this pyruvate cannot enter into Krebs cycle. Now the actual cycle where it have to burn, that means is Krebs cycle. So pyruvate cannot enter into Krebs cycle. So what will happen, because it's a 3 carbon compound, pyruvate will convert into 2 carbon compound, and the name of the compound is acetyl coenzyme A. Now this acetyl coenzyme A will start, it enter into Krebs cycle, it get burned, and due to the burning of this acetyl coenzyme A, right, formation of carbon dioxide and energy takes place, right? And energy is produced in the form of, give me just a minute. So what will happen, that it will acetyl coenzyme may will enter inside the Krebs cycle, and complete burning will take place. Carbon dioxide will be formed and energy will be produced. Energy will be produced in the form of ATP, NADPH, and FADH. And I have already told you that one NADP is equal to, what is that? One NADH is equal to how many ATP?
3 ATP.
Very good. One NADH is equal to 3 ATP, right? You have to remember this formula a second. So one FADH is equal to 2 ATP, right? So one FADH is equal to 2 ATP, and GTP is equal to ATP. Both are the same. GTP, ATP, there is no difference, right? That you have to remember. Okay. So now let's start. First, we will study the glycolysis part, and second, then we will study the Krebs cycle part, right? And you have to remember that glycolysis goes on in cytoplasm, and net gain of 8 ATP takes place in glycolysis. In Krebs cycle, net gain of the 30 ATP takes place, right? The energy which is produced, that is equivalent to 30 ATP, right? So from the one burning of the one glucose molecule when completely burned, the burning of one glucose molecule produces 38 ATP. Right. Now come to the glycolysis part. Right. Try to understand. This is glycolysis. Glycolysis is also known as EMP pathway, Embden-Meyerhof pathway, right? Because Embden, Meyerhof, and Parans, EMP pathway. Embden, Meyerhof, and Parans, these were the 3 scientists who discovered it. So after their name, EMP, it is also known as EMP pathway. EMP stands for Embden, Meyerhof, Parans, right? Pathway. So glycolysis is also known as EMP pathway. Look, we divide glycolysis into 2 parts, right? What are those parts? Look, try to understand. we divide glycolysis into two parts. First part is, we call it preparatory phase. In the green, I have written with the green, preparatory phase. In preparatory phase, first glycolysis absorb, utilize the energy. Means it take the ATP, right? Means it consume the ATP, right? And in the second half part, from here onward, I make this green color arrow. From here onward, this is called payoff phase. Payoff phase, it gives you back ATP, right? In preparatory phase, it take the ATP, and in payoff phase, it give back you ATP. So now let's start with the this one. And I have written over here name Emden, Meyerhoff, and Parans. This was the Gustav Emden, Otto Meyerhoff, and J. Parans. These were three scientists who discovered this pathway. Now, in preparatory phase, first glucose will convert into glucose 6-phosphate, right? One ATP will be utilized. So here I have written that one ATP will be utilized. So this is the minus one ATP, right? One ATP will be utilized. Now, what will happen? Glucose 6-phosphate will convert into fructose 6-phosphate. Again, one ATP will be utilized. So one more ATP loss, right? Now, fructose 6-phosphate changes into fructose 1,6-biphosphate. Till this point, we call it preparatory phase, right? After that, what happened? Fructose 1,6-phosphate breaks down into two molecules, two type of the molecule: dihydroxyacetone phosphate (DHAP) and glyceraldehyde 3-phosphate. Dihydroxyacetone phosphate (DHAP) cannot go ahead in the cannot participate in the glycolysis. Hence, again it convert into glyceraldehyde 3-phosphate. DHAP, that dihydroxyacetone phosphate, and glyceraldehyde 3-phosphate, that G3P, both are interconvertible to each other, right? So every time DHAP again convert into glyceraldehyde 3-phosphate or G3P. Now what will happen? This glyceraldehyde 3-phosphate is this glyceraldehyde 3-phosphate. It convert into 1,3-biphosphoglycerate, and formation of 2 NADH take place, right? It means equivalent to 6 ATP energy is generated here, right? Now, glyceraldehyde 3-phosphate convert into 1,3-biphosphoglycerate. 1,3-biphosphoglycerate again convert into 3-phosphoglycerate. Again, 2 ATP are produced. 3-phosphoglycerate convert into 2-phosphoglycerate, and 2-phosphoglycerate convert into phosphoenol pyruvate (PAP), right? And PEP convert into 2-pyruvate molecule. Again, 2 ATP are produced, right? So that's how the ATP production takes place. Now come to the energy balance sheet. Now here it's energy balance sheet. Look, how many ATP are produced? Look, here minus 2 ATP, because 2 ATP have been consumed over here. I have written minus 1, this minus 1. So total minus 2 ATP are consumed, right? Now 2 NADH molecules are produced, right? So what is the 2 NADH here? 2 NADH molecule produced. So 2 NADH molecule is equal to how many ATP? 6 ATP, right? Again, what happened? 2 ATP are produced here. 1,3-biphosphoglycerate to 3-phosphoglycerate plus 2 ATP. Again, 2 ATP are produced here plus 2 ATP, right? Now I am going to convert them. Minus 2 ATP. Now 2 NADH is equal to how many? Plus 6 ATP plus 2 ATP plus 2 ATP. Net gain is total 8 ATP. So 10 ATP are produced. Out of those 10 ATP, 2 are consumed. So net gain of the glycolysis, 8 ATP. That is the balance sheet of the glycolysis, right? So 2 ATP is consumed and 8 ATP are gained, right? In preparatory phase, 2 ATP are consumed, and in payoff phase, 8 ATP are net 8 ATP are produced. So this is all about the glycolysis. Go through with it and ask me if you have any doubt regarding this. You have to remember the payoff phase, right? How many ATP are produced, how many ATP are consumed. That you have to remember, because you will get the question from here. Energy balance sheet, you have to remember both of Krebs cycle and of glycolysis. Saifullah, is it clear? Enzyme, you need not to remember. You won't get question from enzyme. Did you got it, Saifullah?
Yes.
Alan, Hiba,
Yes, sir.
Aksa, Anifa. Is that clear, glycolysis part?
Yes, sir.
Okay, very good. So definitely you will get the link of this one. So if you want, you can take the screenshot. Now the next one is Krebs cycle. This is the Krebs cycle or tricarboxylic acid cycle, TCA cycle, right? So it is called tricarboxylic acid cycle because the first compound is TCA, tricarboxylic acid, and Krebs cycle. Hence, Krebs discovered it. That's why it is called Krebs cycle. It is also known as citric acid cycle. So that you have to remember all three names: TCA cycle, Krebs cycle, or citric acid cycle. They all are same, right? Look, I told you that glucose will break down into 2-pyruvate, a 3-carbon compound. Now, pyruvate will, when the pyruvate will convert into acetyl coenzyme A, that is 2-carbon compound. So 2 molecule of 2-carbon compound. Then what will happen when it will convert into acetyl coenzyme A? One carbon molecule means carbon dioxide. One carbon dioxide molecule will be lost, and formation of NADH takes place from NAD plus, right? So NADH2 formation will take place, right? Now what will happen from acetyl coenzyme A? This coenzyme A part will be removed from here, right? Now this acetyl coenzyme A will convert into citrate, right? That is a 6-carbon compound. Citrate will convert into isocitrate, again a 6-carbon compound. This isocitrate will convert into alpha ketoglutarate, right? Alpha ketoglutarate. When isocitrate will convert into alpha ketoglutarate, formation of NADH takes place from NAD plus. And again, one carbon dioxide molecule will be formed, right? The reaction will take place in the presence of isocitrate dehydrogenase enzyme. Again, alpha ketoglutarate will convert into succinyl coenzyme A. When it will convert into succinyl coenzyme A, right? That is the third part when the energy is generated. Alpha ketoglutarate will convert into succinyl coenzyme A, that is 4-carbon compound. Again, a carbon dioxide molecule will be released and NAD formation, NADH formation will take place. And the reaction will take place in the presence of alpha ketoglutarate dehydrogenase enzyme, right? Now what will happen? This succinyl coenzyme A, which is a 4-carbon compound, this will convert into succinate, again a 4-carbon compound. Formation of GTP will take place, means equal to ATP. ATP energy will be produced in the presence of succinyl coenzyme A synthase. Succinate will convert into fumarate. Formation of FADH2 will take place. Enzyme succinate dehydrogenase. Fumarate will convert into malate, a 4-carbon compound. Now malate will convert into oxaloacetate, a 4-carbon compound, right? When malate will convert into oxaloacetate, again NADH formation will take place, right? Now again the oxaloacetate that will convert into acetyl coenzyme A, right? So that's how the reaction will keep on going, right? And the complete burning of the acetyl coenzyme A or glucose takes place. Now let's calculate where the energy formation takes place. Here formation of NADH takes place. Look, you can see. Here the NADH2 will be formed. Here NADH will be formed, right? Again here NAD plus will be formed, right? So let's calculate how many NADH are formed, right? How many NADH are formed? Let's start. Look, one NADH here, one NADH here, one NADH here, right? 1, 1, 2, 3, 4. To total, how many NADH will be formed? You can see in this balance. This is the energy balance. So formation of, sorry, just a minute. Okay, this is the reaction. Glucose. Let's forget the reaction. Don't go to the reaction because that will become difficult for you. It is not required, right? So how many? Look here. Formation of in the Krebs cycle, 2 ATP are produced. Mean 2 GTP is equal to ATP. So 2 ATP are produced here. You can see 2 ATP are produced here. 2 GTP are produced means 2 ATP are produced. And here At every place, 2 NADH are formed. 1, 2, 3, 4. So total 8 NADH are formed. I have written over here. Total 8 NADH2 are formed. 8 3 are 24. So 24 NADH. 2 FADH2 are formed. So that is equal to 4 ATP. So 2, 24 plus 4, total 30 ATP are produced. Equivalent energy is produced in the Krebs cycle. In glycolysis, we have gone through with that, and we know that the 8 ATP are produced. So 8 plus 30, 38 ATP are produced in the Krebs cycle, right? So during the respiratory process, 38 ATP are produced from one glucose molecule. That I have told you, and if you get the question that somebody was asking a question, like either the answer will be 36 or 38. So this is proof that there will be 38 ATP formation will take place in the burning of one glucose molecule. What you have to remember? You have to remember the number of the ATP, number of the NADH, and number of the FADH2 are produced. So that's why I have made the balance sheet, energy balance sheet for you. And normally you won't get the question from the enzyme, but yes, this is the simplest cycle which you need to remember. But you have to remember this one at any cost. You have to remember this one, because if you won't remember, you will forget it. Now tell me, any one of you, if you have any question, any doubt, please ask me. You have to remember this cycle. You have to remember the glycolysis, and then energy balancing. These things you have to remember. These are the only questions in. Ask me if you have doubt.
So few steps are not there in textbook.
In the NCERT book.
Yes.
Yes. So that's why I have represented this cycle. You have to remember this cycle. You know, from the NCERT, you won't be able to catch those questions. You will see that, because it's very simple kind of a cycle. Yeah, this you have to remember minimum this cycle. There are number of the cycle which are much more complex. If you see the Pradeep, Truman, ABC, A to Z, other books, right, Arihant, there are very complex kind of the cycle, but this is the simplest and sufficient for your medical examination.
Okay, sir.
Fine. Wherever the carbon dioxide molecule is going out, those are called decarboxylation. Look, here the carbon dioxide is removed. Here carbon dioxide is removed. This is decarboxylation. This is decarboxylation. This is decarboxylation reaction, right? Any doubt? No?
No, sir.
Well, come to the electron transport chain. This electron transport chain is almost similar to the electron transport chain which we have seen in the photosynthesis part. Look, even here, the same thing you have to remember that it takes place. Now, one more thing I want to tell you. Since I have started the photosynthesis, I am talking about this one, that 1 NADH is equal to 3 ATP, 2 FADH is equal to 2 ATP. How? Let me prove it. How does it take place? The formation of ATP takes place in the same way which I have told you in the electron transport chain of photosynthesis, like in chloroplast. The same way it takes place. Their energy used to be generated in the chloroplast, but here the energy generation is taking place in mitochondria. That is the only difference. Even here, the same like the chloroplast, at the leakage of two hydrogen ion, 1 ATP is produced. Look. Now I have told you that in the citric acid cycle, means Krebs cycle, the formation of NADH, NADP, formation of NADH2, FADH, and GTP takes place. So GTP is itself ATP, right? But how the NADH2, NADH2, and FADH2 are the energy currency. Look, that NADH will break down into NAD plus hydrogen. When this NADH will break down, what will happen? The formation of hydrogen ion will take place, and the electron will be produced. When now it will go to this is the inner membrane of the mitochondria. Look, this is the outer membrane of mitochondria. This is the inner membrane of mitochondria. I have written over here, outer membrane, inner membrane, and there is a space between these two membranes. This is called intermembrane space, right? and in inner membrane there are certain cytochrome complex. Cytochrome complex first, third, fourth. Here first, second, third, fourth. Right. Now when NADH will break down, NADH will break down into NAD plus hydrogen ion. Now the electron will pass through cytochrome 1. When electron will pass through with the cytochrome ion, it will pump out 2 hydrogen ion in the intermembrane space. Now the electron will pass to the quinine. Quinine will take electron to the cytochrome third. When it will reach to the third, it will again pump out 2 hydrogen ion. Now it will pass to the cytochrome C. Now when it will pass to the cytochrome 4, what will happen? Again, it will pump out the 2 hydrogen ion. So total 6 hydrogen ion are pumped in an intermembrane space. Now finally, the electron will be taken by oxygen molecule, and the formation of water molecule will take place. Right. So water is the final acceptor of the electron. Now look here, total due to breakage of one NADH molecule, 6 hydrogen ions are pumped in. Now these hydrogen ion cannot leak out. Now there is a potential gradient or hydrogen ion potential gradient between inner membrane and outer membrane, the inner part and outer part. In intermembrane there is a high hydrogen ion concentration. In matrix there are low hydrogen ion concentration. These hydrogen ion will leak out inside, and when these hydrogen ion will leak out, formation of ATP will take place. When 2 hydrogen ion will leak out, how many ATP will be produced? 1 ATP. When 6 hydrogen ion will leak out, how many ATP will be produced? 3 ATP. That's why 1 NADH2 is equal to 3 ATP. Is it clear or not? Keep on telling me if you are
Sir,
getting
can you
my.
explain again?
Yeah, sure. Look, I have told you that, look here, 1 NADH2 is equal to 3 ATP. How 1 NADH2 is equal to 3 ATP? Actually, when 1 NADH2 breakdown, formation of 3 ATP takes place. How? Look, 1 NADH2 breakdown over here. Now the electron passes through complex 1. It have pumped in 2 hydrogen ion. Means 1 ATP will be produced. Again, this electron will pass through the third. 2 hydrogen ion more will be pumped. Again, it will pass to 4. 2 hydrogen ion will be pumped. So due to passage of electron, how many hydrogen ion are pumped? 2 plus 2 plus 2, 6. So when 1 NADH will break, how many hydrogen ion will be pumped in the intermembrane space? 6. Now when these 6 hydrogen ion will leak out, how many ATP will be produced? At the leak out of 2 hydrogen ion, 1 ATP is produced. At the leak out of the 6 hydrogen ion, how many ATP will be produced? 3 ATP. That's why 1 NADH2 is equal to 3 ATP. Now is it clear? Clear?
Yes.
Okay. Now why 1 FADH2? This one. Why 1 FADH2 is equal to 2 ATP? Look. This FADH2 will break down into FAD plus. When FADH2 will break down into FAD plus, what will happen? Hydrogen ion will be pumped in. Where? Now cytochrome complex third is not going to pump in the hydrogen ion. Why? Because it do not traverse through this membrane, right? So what will happen? It will pass electron to complex third. When the electron will reach to the complex third, 2 hydrogen ion will be pumped in. Now this electron will reach to the fourth, 2 hydrogen ion will be pumped in. So due to breakage of 1 FADH2, how many hydrogen ion pumped in? 4 hydrogen ion. At the leak out of 2 hydrogen ion, 1 ATP is produced. So when these 4 hydrogen ion will be leaked out, how many ATP will be produced? That's why 1 FADH2 is equal to 2 ATP. Is it clear?
Yes, sir.
So this is the electron transport chain of the or electron transport system of the micromolecule. But the more important electron transport system is important in the photosynthesis. Here there are 2 things more important. That is glycolysis and Krebs ring. That you have to, right? Here hardly you will get the question from electron transport system. You may get, but that electron transport chain is more important. Clear?
Yes, sir.
Hanifa, is it clear?
Yes, sir, it's clear.
Now, what is amphibolic pathway? It is given in the last of your NCRT as well. This is the amphibolic pathway. Look, it is important. Rarely, but I have seen the question from here as well. Look.
Sir, can you go to the other one last?
That one, electron transport system.
Yes, sir, done.
Look, now what is the amphibolic pathway? In amphibolic pathway, I have told you there can be substrate can be of three types. It may be carbohydrate like starch, it may be fat, it may be protein. Look, when the carbohydrate breaks down, so we have normally the substrate is the carbohydrate, means sugar. So starch break down to glucose, right? Glucose. Glucose will convert into glucose 6 phosphate in glycolysis, fructose 1,6 biphosphate, DHAP, then glyceraldehyde, G3P, then pyruvic acid. Pyruvic acid will convert into acetyl coenzyme A, and acetyl coenzyme A will go into Krebs cycle and generation of energy will take place. Right? Okay. Again, so that we have studied in detail. But fat is also source of energy and protein is also source of energy. We have taught in the starting of the chapter that all these three can be substrate, right? Means they can burn. Now, if protein will burn, protein will break down into amino acid. Amino acid will convert into pyruvic acid. Pyruvic acid into acetyl coenzyme, and again acetyl coenzyme will enter into Krebs cycle. If this is fat, fat will break down into fatty acid and glycerol. Fatty acid will directly convert into acetyl coenzyme and will go into this cycle. Now, if this is glycerol, glycerol will convert into dihydroxyacetone phosphate. DHAP will convert into G3P. G3P will convert into pyruvic acid, and pyruvic acid into acetyl coenzyme, and then it will burn. So this pathway is called amphibolic pathway. Clear? Now there is a question that you must have encountered this question in your 11th class examination. That this pathway, Krebs cycle, is catabolic pathway or anabolic pathway. Let me recall you. Catabolic pathway are those pathways which are destructive pathway, where destruction is taking place. So breaking down is fat taking place, breaking down is carbohydrate taking place, breaking down of the protein is taking place. That is catabolic reactions, where something is getting broken, right? Anabolic reactions are those which are synthetic reaction, where construction is taking place, synthesis or formation of something taking place, right? So normally it looks like that Krebs cycle is catabolic pathway. No. Krebs cycle is both catabolic and anabolic pathway. That's why we call it amphibolic pathway. Look, why? Krebs cycle breaks down everything, right? It breaks down the fat, it breaks down the carbohydrate, the protein. That's why it is catabolic. But for during the Krebs cycle, formation of acetyl coenzyme A takes place. Acetyl coenzyme A is the raw material for the synthesis of fatty acid, steroid, turpentine, carotenoid, and some other aromatic substances. That's why synthesis is also taking place and breaking down is also taking place. That's why we cannot say that Krebs cycle is a catabolic pathway. It is both catabolic as well as anabolic. And those pathway which are catabolic as well as anabolic, they are called amphibolic pathway. Hence, Krebs cycle is an amphibolic pathway because both catabolic and anabolic reaction goes on in the Krebs cycle. Tell me, is it clear or not?
Yes.
One last, that is the anaerobic pathway. This diagram is there in your NCERT. Look, irrespective of the reaction, either it's aerobic or anaerobic, whatever it is, right? In both kind of the respiration, what happened? Glycolysis goes on. That is for sure. So first thing you have to remember that glycolysis goes on for both. Either it's aerobic reaction or it's anaerobic reaction. So look, glucose, that is glyceraldehyde 3-phosphate. Glucose break down into glyceraldehyde 3-phosphate, then phospho, then again phosphoenol PEP, pyruvate, then pyruvic acid. So we know this is the glycolysis and every reaction. So in glycolysis and this one, both 8 ATP will be produced. That is for sure. Now there are two fate of the pyruvic acid. Pyruvic acid, if this reaction is taking place in bacteria or yeast, what will happen? Pyruvic acid will break down into ethanol, means ethyl alcohol, and carbon dioxide, and formation of NAD and NADH will break down into NAD plus, right? So in ethyl alcohol formation takes place in yeast, and lactic acid formation takes place in bacteria. Look, pyruvic acid will break down into acetaldehyde and carbon dioxide. Acetaldehyde will convert into ethyl alcohol and NAD plus, right? This reaction goes on in the yeast. In bacteria, pyruvic acid plus NADH and formation of lactic acid plus NAD plus will take place, right? And again, in bacteria do not require oxygen, right? That's why the respiration is anaerobic, and fermentation also takes place is anaerobic reaction. Bacteria produces ethanol, that is C2H5OH, and lactic acid. The same reaction, this formation of lactic acid takes place in human muscles as well during the time of the fatigue, right? When we do heavy exercise and or we run fast, then what happens? The anaerobic respiration start, and the formation of lactic acid takes place in our muscles as well, right? So what happened with the lactic acid? What happened with those lactic acid? Will deposit in our muscles? Can anybody tell me? When you run fast, your muscles require more oxygen, but oxygen cannot be supplied in that frequent, fast manner. So it do not matches with the pace of the supply. Do not matches with the pace of the demand. So what happens? Some of the muscles start anaerobic respiration. Then deposit formation of lactic acid takes place, and lactic acid deposit between our muscles. So lactic acid, due to presence of lactic acid, we feel muscular cramp. When we take the rest, this lactic acid goes to the liver, and it is again converted into pyruvate, right? Is that clear? Anaerobic respiration.
Yes, sir.
Okay. So this is all about the respiration in the plant. Go through with your NCERT. If you have any question, please ask me. And if you want me to dictate anything, then I'm ready to dictate. Go through with this PPT. And if you want to ask anything, right? Most of the thing they because it's exhaustive, right? Krebs cycle to it takes lots of energy to read the Krebs cycle and to remember it, and this you have to take the pain to memorize these lactic this one anaerobic respiration, aerobic respiration, and glycolysis. And some of you leave this chapter, and then they pay for it during the examination, because if you leave this chapter, you have to pay during your NEET examination. So don't leave. What you have to do, you have to remember these two cycles. Make these two cycles today only. After the class, give 15 minutes. Make these two cycles in the A4 blank sheet. Paste it in front of your table where you study. Watch it every day. You'll memorize it within a week, right? And you'll learn it. It will imprint it in your brain, right? So you have. So in case if you have any doubt, any question, please ask. Any one of you. Let me ask you one by one. Aksa, is it clear? Everything clear? Mamuna,
Yes, sir.
Mamuna, Farhat, Hiba, Ellen, Hanifa. If you want any kind of the help in this chapter, please ask me. Feel free to ask me anything. So what you have to remember? Glycolysis, Krebs cycle, anaerobic respiration, energy balance sheet of glycolysis, energy balance sheet of Krebs cycle. That's it. That's what you have to remember. Now, what is the next chapter? Next one is respiration. Plant growth and development. Here you will get the question from hormones only. Plant growth and development. Auxin, gibberellin, abscisic acid, all are important. Plant growth and development. Yes, this is it. Yes, these concepts are also important. Very, very important. What is de-differentiation? What is the redifferentiation? Can any one of you tell me what is the redifferentiation? Any one of you? What is redifferentiation? Or what is de-differentiation?
When a cell gets a particular function.
That is called differentiation.
You're asking de-differentiation. When it loses the function.
Suppose that there are 10 people, 10 students. They all are the same student. They have not been given any particular job. So that is the call them meristematic student. They do not have any specific job, right? Now, in front of me, they all students, they're 10 students. I ask one of them to make the question paper, another to teach the student, and another to check the copy, another to inspect. I have given particular job to every student. They have their particular job. Those who are making the question paper, they will do it this job only. Those who are teaching, they will do this job only. Right. I have differentiated all those students. Right. Now they cannot be neutral. They cannot go to previous stage. So this is the differentiation. When a particular cell is assigned a particular job, that you have to do this one only. This is differentiation. Now they cannot come to their original stage, right? Now there are some students who are teacher. I want them to do some other job. I want them to print some book. So what I will say, I will again taken them back as a neutral position, right? I have like removed from that job and take them back in my seat as a neutral student. This is called de-differentiation. Their job have been removed. Now again, I have given them another job to go and print the copy. Now this process is called redifferentiation. Why? Because they were already in the job. So those who have been already defined a particular job, and they are taken back again, they have assigned a new job. That is called redifferentiation. Got it? Look. Now there are certain cells, the new cells which are formed at the top of the plant or at the top of the root, they are called meristematic cells. Meristematic cells are very soft cell and they have capability to divide, right? They divide in very fast manner. Their cytoplasm is very dense. They have a small nucleus and no vacuole. That is the property of meristematic cell. But they divide very fast. Now these meristematic cells have given a particular job. they become a fully mature cell and opt a particular function and loses the property of division. Their division property loses. They are called differentiated cell. And this process is called differentiation. And every cell have been assigned its job. Either it's have to do photosynthesis, or it's have to store the food, or it have to conduct the water. Now what happened that these differentiated cell, right? Now it's a part of the leaf, or it's a part of the stem. Now the stem or leaf that get damaged. Now what will happen? The cells have been assigned particular job. They cannot divide. Again, what happens if damage takes place? These cells again regain the capability of division. This is called de-differentiation. They have been assigned, they lose the capacity of division, but again they have got the capacity of division. This is called de-differentiation. It regain the property of division and start dividing, right? Start dividing, and cell become meristematic and divide. Again, now these meristematic cell, they have been assigned a particular function, right? When these cells, de-differentiated cell, assigned a particular, again differentiation takes place. This is called redifferentiation. Again, these cells get its function and stop dividing. This is called redifferentiation, right? Look, what happened in the vascular bundle? There are interfascicular cambium, right? So these interfascicular cambium, that is the meristematic cell. But between these vascular bundle, there are intrafascicular cambium. Formation of intrafascicular cambium takes place from normal parenchymatous cell, right? So this process is called formation of interfascicular cambium takes place due to de-differentiation process, right? Again, they will divide and form secondary xylem, secondary phloem. That we will call that redifferentiation. Tell me, is it clear or not? If it is not clear, then ask me. Come on, guys, tell me. You will get the question. Right? Yes, I have seen the question from de-differentiation, redifferentiation, and all that. Formation of interfascicular cambium is de-differentiation, differentiation, redifferentiation. So that question was there. I have seen that question. That's why I have made this diagram. So formation of interfascicular cambium is de-differentiation process. Is that clear?
Yes, sir.
What is the plasticity? Okay. First, let me plant growth curve. Let me tell you the plant growth curve. This is sigmoid curve, and in the chapter of ecology, ecosystem chapter, we have just a minute. Give me 1 second. Look, uh, this is the sigmoid growth curve, right? So this is the axis of the time and growth or size or weight of the organism, right? So that's how the growth takes. Initially, the growth is a bit slow. This is called lag phase, right? Then what happened when it becomes juvenile, right? The plant grow very fast pace. That is called log or exponential phase. At the point of maturity, when it will start bearing the flower and fruit, at the point of maturity, it will become stationary, right? Further growth do not take place. Now this is called stationary phase. So first phase is lag phase, when initially in initial days the growth is slow, and then what happened, then it gets a log or exponential phase and the growth is very fast. At the maturity, the growth will stop, or it will become very slow, and after some time, after maturity, it will become stationary growth. And when it will complete its any organism, when it will complete its uh, life span, the senescence or death will take place, right? What is the plasticity? Can anybody tell me what is the plasticity? No? Look, different habitat and in different environment, the part which changes their shape and to adopt the environment, that's this property is called plasticity, right? Look, this is the leaf of larkspur. When the larkspur is found in terrestrial habitat, means land, their leaf will be like this one. When they will be juvenile, their leaf will be like this one. When they are adult, their leaf will be like this one, right? So in juvenile stage, the larkspur leaf structure is different. In adult stage, its structure is different. If larkspur is found in the terrestrial habitat, the leaf like this way. If it is found in the water habitat, the leaf will be like this way, right? So in different phase of the life and in different habitat, the same plant species changes its structure, right? It's a structure. This property is called plasticity, right? Right? And this is example of heterophyly. Heterophyly means different type of the leaf, same plant but different type of the leaf is called heterophyly. So heterophyly is the example of plasticity. Is it clear or not? Tell me, guys, is it clear or not?
Yes, sir.
Clear? Very good. You people are very smart one. Plasticity have been asked. Plant growth regulator, right? Plant growth regulator. So there are different kind of the plant growth regulator. So basically they are the plant hormone. So plant hormones or plant growth regulator, same thing. They are also known as the phytohormone, right? So plant hormone, phyto means plant. Plant hormone are also known as the phytohormone. So there are only 5 kind of the plant hormone, because plant body is very simple kind of the body. Human body or animal body is very complex body. But plant body is very simple kind of the body. So there are different kind of the hormones. So phytohormones, they can be auxin, gibberellin, cytokinin, abscisic acid, and ethylene. These are the 5 plant hormones. What is the function of plant growth regulator? Growth, differentiation, and development. Growth of the plant, differentiation of the cell, and development of the cell. Plant growth regulator, they differ from one or they differ from one organ to another organ. Actually, their function, like the auxin. The auxin will perform different function in the root, different function at the shoot, and different function in the leaves, right? So their function differ from one organ to another organ, right? Similar response may also be seen in different plant growth regulator. Different plant growth regulator can perform the similar function, right? They are required in low concentration, right? And site of production are they are maybe different site of production and different site of the action. Means they are produced at different part, they act at different part. Like auxin, it is produced in the tips of the growing tips, but they perform their function at different parts, right? So that is the function of plant growth. Is that clear? Now let's take function one by one. Auxin. Look, the main function of auxin is that you must have seen the sunflower. Sunflower, what is the property of sunflower? Tell me, anyone of you. What is speciality with the sunflower?
It face towards the sun.
Right, that's why it is called sunflower. If in the morning you will say it, they all will be, they are faced toward the east direction. And as the sun move, their faces will move. In the evening time, it will see all the sunflower will have its mouth toward the face toward the that is the south direction, right? So what happened? How they move toward the direction of the sun? Look, oxygen is produced at the top. When the sun rays fall like this way, oxygen get accumulated in the opposite direction. So oxygen, sun rays is falling at this point, but oxygen get accumulated in opposite point. Where the oxygen accumulate, it elongate the cell. So when it will elongate the cell, pressure will be created like this way, this direction, and they will cause the bending of the this part. And that's why the sunflower always have their face toward this, right? So oxygen is responsible for cell elongation, and this type of the movement is called phototropic movement. When movement in the direction of light takes place, this is called photo means light, tropic means movement, right? In the directional movement. So when the movement takes place in the direction of the light, it is called phototropic movement, right? So what is the first function? First and foremost function of the oxygen is phototropic movement. First thing. Second, cell elongation. It is responsible for increasing the length of cell. So cell elongation. First, it is the first discord. This is very, very, very important. These all questions are very important. You have to just cram all these things. First discovered oxygen was indole butyric acid, IBA, right? Indole butyric acid. Natural oxygen, that question, these both questions have been asked in NEET. Natural oxygen, indole acetic acid. Chemical, what is the chemical name of oxygen? IAA, indole acetic acid. So first discovered oxygen was indole butyric acid. What is the natural oxygen? IBA and IA are the natural oxygen. It was discovered by Clogg et al. Clogg et al. Discovered it. Discovered oxygen, right? They discovered oxygen. Synthetic oxygen. What are the synthetic oxygen? They are the unnatural oxygen. They are man-made, factory-made oxygen. What are synthetic oxygen? This question has been asked. Both 2,4-D and 2,4-T. 2,4-D, 2,4-T, and NAA, right? NAA. These are the synthetic oxygen. 2,4-D stands for 2,4-dichlorophenoxyacetic acid. 2,4-5-T, 2-trichlorophenoxyacetic acid. NAA, naphthalene acetic acid. Function, they stimulate respiration, cell enlargement, xylem differentiation, cell division in the cambium, means secondary division, secondary growth, root initiation. These both where I make the star, they are the questions. Apical dominance. What is the apical dominance? Oxygen is responsible for apical dominance. Means the plant will grow in the length, but it will suppress the horizontal growth. It will increase, means promote the vertical growth and suppress the horizontal growth. That is called apical dominance. Phototropic movement, I have told you. And female flower in some plant, it is responsible for formation of female flower in plant. Means if you spray the oxygen, it will promote the formation of female flowers. Tell me if you have any doubt in all this. Is that clear?
Yes.
You have to remember all the points which I have explained. All the points. They all are questions. You will find this question in previous years. Is it clear? Saifullah, clear?
Yes, sir.
Memuna, Farad, clear?
Yes, sir.
Now the function of gibberellin. Gibberellin is another hormone. The property of gibberellin, that is, this is weakly acidic. So it is weak acidic. Function that it increases the internodal growth. So these are the node, like node of bamboo, node of sugarcane, right? So this is the internode. This part is called internode. So it is responsible for increasing the internodal growth, right? So internodal growth is increased by this one. These part are called internodes, right? They are the internodal. So they increase the internodal length. So if we'll spray the gibberellin in the sugarcane, what will happen? They will increase the internodal length, means they will increase the productivity of sugarcane. So they are very important for the sugarcane, right? These kind of the crops. Okay. So and these part are called nodes. These part are basically called nodes. They are called. And nodes are the part from where the leaves or the branches originate. New leaves and new branches originate, right, from the node part, right? So these are called nodes. Now, what happened? It was discovered by Kurosawa and Harry, right? Uh, where it was discovered. So, who was the Kurosawa? Kurosawa was a farmer. He was not a scientist. He was a farmer. So, Kurosawa was a farmer, and he discovered. Where he discovered this? This is discovered in a fungus, and the name of that fungus Gibberella fusakurai, right? So, Gibberella fusakurai is a fungus where gibberellin have been discovered. This fungus causes a disease that is called Bacchanae disease. Bacchanae disease means foolish seedling, Bacchanae disease of rice. This fungus causes Bacchanae disease of rice, and this is also known as foolish seedling disease because there will be seedling, but you will not found inside any rice seed, right? So this is called foolish seedling disease, and here in this fungus it was discovered. What are the functions? Look. Growth in stem, leaf, and aerial part means growth of the shoot. It causes bolting. What is bolting? Bolting is the process where genetically, what is that? They increase bolting. What is it? Bolting increases the height of increases the height of genetically dwarf plant. Right? Look, this was a cabbage like this way. Cabbage, inside the cabbage, there are very small internode, and from the nodes, these leaf come out. If you will spray gibberellin, the cabbage will become 5 feet tall. You can see this is internode. It have increases. The cabbage has, this is the cabbage. This is the real picture of the cabbage while spraying the gibberellin. So it increases the length of genetically dwarf plant. This process is called bolting, and bolting is done by the gibberellin. This question have already been asked many times, and it even again it can come, right? It overcome the natural dormancy of birds, tuber, and seed. Means it overcome with the dormancy. Suppose that there is a dormant seed, means it is not germinating. Spray the gibberellin or soak with gibberellin, they will start germinating, right? So overcome the bird, tuber, and seeds. Flowering in long day plant, LDPs, right? So it is responsible for falling and promote the male flower, right? They do not promote. So oxygen was responsible for promotion of female flower, and this is responsible for promotion of the male flower. Is that clear? Is it clear to all of you? Keep on responding me, guys. Then I will come to know that either you're
Yes, sir.
getting it or not. Okay, fine. So cytokine. cytokinin is basic in nature, right? This is basic in nature. Just a minute. Okay, look, this is basic in nature. It was discovered by Skoog and co-worker who discovered it. Promote the cell division. Cytokinin is responsible for cell division, right? Basic function is cell division. Like basic, if you'll ask me the basic function of oxygen, that is uh, that is also responsible for phototropic movement, right? Cytokinin, cell division. Zimberlin, growth. So what is the function? Cell division, cell elongation. Oxygen plus cytokinin causes morphogenesis. What is the morphogenesis? Tell me, guys, what is morphogenesis? Morphogenesis means that is formation of new organ and cell differentiation. That is called morphogenesis. Formation of new leaves, formation of new stems, formation of new branches. That is called morphogenesis, right? Formation of new organ. So when oxygen and cytokinin both work with each other, they cause morphogenesis. They delay the senescence. They delay the senescence. Senescence means death. Suppose that if the leaves of the particular beautiful plant are dying, right, or they are falling, a defoliation is taking place, right? If you spray the oxygen, they will stay for the longer time period, right? They overcome the seed dormancy. Again, they remove the seed dormancy. They provide resistance to the temperature. They indicate parthenocarpy. Parthenocarpy means seedless plant, right? One more thing. In tissue culture, when there is a mass of the cell, right? In the mass of the cell, that is called callus, right? Callus. These callus is what is the callus? They are undifferentiated cell. We supply the oxygen and cytokinin in some ratio. What happens? Formation of root and shoot takes place. Means morphogenesis start, right? Cell differentiation start. So it is used in the tissue culture as well. Tell me, guys, is it clear or not? Cytokinin. Mamuna,
Yes, sir.
Hiba, Farhat, clear?
Yes, sir.
Great. You will 100% get the question from these hormones. Believe on me. Again, ethylene. Ethylene is the
Sir, can
only
you go back,
gaseous
please?
hormone which is present in biological world. No hormone is found in the gaseous state in even any plant or animal. This is the only gaseous hormone. It stimulate the transverse growth, means horizontal growth, like this growth in this fashion. Maximum senses occur during a ripening of fruit. When the fruit get ripened, then formation of ethylene takes place. It is responsible for ripening of fruit, right? Ripening of fruit. You know, nowadays what we are doing. Nowadays, suppose that you have to ripen the banana. You have the one truck banana. A complete room is filled with the banana. There is an airtight room, airtight chamber, where they are an ethylene cylinder. They open the cylinder like deodorant. They open the cylinder. They throw the cylinder. Ethylene keep on leaking from those cylinders, right? And they close down that chamber. In the morning, every fruit get ripened, right? They get the ripened banana. That's how the ripening is done, right? So they are responsible for ripening of the fruit. They hasten the senescence of leaves, right? If you will spray the ethylene, they will hasten the seed, right? And again, they breaks down the dormancy of the seed, right? they increase the length of the rice. They are responsible for root initiation. That is important. They are responsible for root initiation. They stimulate flowering in the pineapple and mango. This question I've been asked multiple times. Multiple times. Pineapple question. This multiple times this question I've been asked. Right. So ethylene is responsible for ripening of mango. I think hope it should be clear. It's very simple. Abscisic acid, ABA. Abscisic acid, it is known as the stress hormone. This question has been asked. Why stress hormone? It prevent or protect the plant at the scarcity of water. Protect plant at the scarcity of water. The scarcity of water, or in drought. What happens? Suppose that there is a scarcity of water. The plant is about to die. So what will happen? The formation of abscisic acid will take place. What abscisic acid will do? Abscisic acid will close down the stomata and increase the rate of the transpiration. By this way, it will increase the rate of it will stop the rate of, it will close down the stomata, stop the rate, decrease the rate of transpiration, so that the plant could survive for the longer time. Right. That's why it is called stress hormone. It is also called dormant. Why? Because it is responsible for dormancy of the seed. Right. If abscisic acid is present, seed will keep on sleeping. Right. It will remain dormant. Right. So it will called dormant because induces dormancy of seed. Dormancy, is it clear? What is the dormancy? Tell me, guys, is it clear or not? Dormancy clear or not? Tell me.
Yes, sir.
Okay, very good. Abscission of the flower and fruit. What is the abscission? The ripened fruit and flower, they fell down. When a leaf become yellow, it fall down. When fruit become like ripen, it fall down. How does it happen? Suppose that when ripening of or where leaf become ripen or mature, right? Formation of dead tissue will take place. That is called abscission layer. Formation of abscission layer. What is the abscission layer? Formation due to the death of tissue, right? The dead tissues are accumulated over here. That is called formation of the abscission layer, and the leaves fell down, right? Again, if the fruit get ripened, right? Due to abscisic acid, formation of abscission layer will take place. Abscission layer is again the a layer of dead tissues with the form, and fruit will fall. That's also falling of mature leaves, falling of ripened fruit takes place due to the what? Abscisic acid. It reduces the transpiration. Again, a very important question, responsible for flowering in the strawberry, right? Abscisic acid responsible for flowering in the strawberry plant, right? Is that clear?
Sir, can you show cytokinin again?
Yes, I'll show you. Just a minute.
Yes, I done.
Yeah. Now, what is the photoperiodism? Photo means light, period means duration, is means impact. So, impact of duration of light is called photoperiodism. I have broken down this word, photoperiodism. So, photoperiodism is nothing but impact of light duration, right? Very important question, where it was discovered, right? So Garner and Allard discovered in a tobacco plant. This is not Nicotina tobaccum, guys. Remember, this is not Nicotina tobaccum, because normally tobacco which is eaten and which is smoked, that is called Nicotina tobacco, right? That is not. Garner lard discovered in a tobacco plant, and the name of that is a wild tobacco plant. Name is Maryland mammoth. So Maryland mammoth is the name of wild tobacco plant. Here it has been discovered. Photoperiod. There are three types of the plant: short day plant, long day plant, and day neutral plant. Look, what are the short day plant? They are the plant, they generally flower at the winter time, right? They flower at winter, means reproduction, flower at winter or autumn, right? Why? Because in the winter, there is a very small day length, right? Light remains for a small time period, right? So in the winter, days are shorter, means photo period is shorter, right? So in winter, they are the xanthium. You have to remember the name, like example, xanthium, chrysanthemum, dahlia, rice, sugarcane, strawberry, potato, and tobacco. The example which is written over here, you have to cram it by heart because you will get the question from which of the following SDP, which of the following is LDP, and which of the following NDP, right? DNP. So now long day plant. Long day plant normally they flower and summer. They flower at summer point. Why in summer? Because at summer time the day photo period is longer because day length is longer, right? So they are called long day plant. Henbane, wheat, oats, sugar beet, radish, and barley, right? Day neutral plant. They are called DNP, day neutral plant, which are not affected by the long day or short day. All 12 months they flower. They can reproduce at any point of the month, right? So tomato, pepper, cucumber, pea, sunflower, maize, and cotton. They are day neutral. So on the basis of light exposure, this plant have been divided into three categories: SDP, LDP, and DN. Clear? Will you remember these names? You have to actually, if you want to crack the question. Last but not the least, what is the vernalization? What is the vernalization? Any one of you? What is the vernalization? Come on, guys, tell me what is the vernalization? Who is going to tell me? No one. No one is going to tell me. Come on, amazing. I'm shocked. Saifullah, vernalization. Maimuna,
Don't know, sir.
look, if suppose that any plant, suppose that any seed is not germinating, right? Or its lifespan means flowering is delayed. It takes 6 weeks to flower, 16 weeks to flower, means 16 weeks to reproduce, right? Before sowing that plant, before sow into soil, we treat with the low temperature, right? We give a temperature shock of 0 to 5 degrees centigrade, right? So due to low temperature, then high temperature, then low temperature. So we give them temperature shock. So before sowing that plant, if we'll sow that plant, their reproductive cycle or reproductive lifespan will reduce, right? They will fly in the 6 week only. This process is called vernalization. So vernalization is giving the chill treatment or low temperature to the plant, to the seeds or seedling before we sow it, right? That's this process is called vernalization. Is that clear? Vernalization part.
Sir, can you explain again?
Yes, suppose that I have a plant. I am going to sow it in the soil. It will germinate and it will give flower and fruit after 16 weeks. Now I want to reduce this time period of 16 weeks, because if I will reduce, then I will get the fruits and seed earlier, and then my field will be free to sow another crop, right? So I will give it chill treatment. I will keep it in freezer, right, where the temperature is 0 degrees centigrade over the night. Then after freezing that, I will sow the seed in next morning, right? I will soak in the water and then sow in the next morning. Then what will happen? Now the flower, that seed will germinate and it will flower and give the fruit within the 6 weeks, right? So by this way, I have reduced the production time, or I have reduced the life span or flowering time of the that particular plant. This process of giving chill treatment before sowing into soil is called vernalization. Clear?
Yes, sir.
And what is the temperature for vernalization? 0 to 5 degrees, right? So we have completed this chapter as well, and go through with the NCERT. If you have any doubt, please ask me now. Is that clear? Or it's okay? This was the last chapter of plant physiology. We have completed the plant physiology.